Escherichia coli is a rod-shaped bacterium that is normally found in the intestines of the human digestive tract. It has similar organelles to most other bacteria, including a cell wall, a cell membrane, cytoplasm, ribosomes, a nucleoid, pili, flagella, and plasmids. It uses deoxyribonucleic acid as its genetic material in the linear form contained in the nucleoid. E. coli replicates with a method known as binary fission, where the cell replicates interior structures and divides into two new cells. It also exchanges plasmids with others of its kind. Certain plasmids can cause E. coli to become virulent, creating enterotoxigenic E. coli that can infect our gut and cause gastrointestinal problems. Plasmids can also be used to engineer these bacteria to perform tasks humans want them to do. Several researchers have explored E. coli's potential in metabolic engineering, specifically in using its unique pathways to turn renewable materials such as corn and grass into bioethanol. Bioethanol is a type of fuel that is able to be used in vehicles such as cars. This type of fuel is revolutionary because it is renewable. Since humans have been growing corn and grass for millennia, we are capable of producing enough of the materials needed to power today's vehicles. E. coli and other microorganisms such as yeast are used to ferment the input materials such as corn and grass into bioethanol. Specifically, lignocellulosic feedstock material, which is composed of parts of plant cell walls, is used as the main material to create bioethanol using E. coli. As seen in the research paper Role of Escherichia coli in Biofuel Production, the production of ethanol can be carried out through the chemical reaction of glycolysis. Figure 1a displays the strategy for the generation of bioethanol through a glycolysis process within E. coli. Ultimately, the redox reaction for the reduction of acetyl-CoA into ethanol by aldehyde alcohol dehydrogenase, which is an enzyme that catalyzes this reaction to ultimately produce ethanol, results in an input of glucose, leading to an end product of the biofuel ethanol, as well as a byproduct of acetate. However, in a natural environment, the ethanol produced within this reaction is then used for carrying out the regular functions of the cell, and there's also an unwanted byproduct of acetate. This process also creates a suboptimal amount of biofuel, as 0.26 grams of ethanol per gram of glucose is not anywhere near the theoretical maximum yield of 0.51 grams of ethanol per gram of glucose for an E. coli bacterium. Thus, genetic engineering was used to insert PDC and ADHB genes from a different bacterium, Zymomonas mobilis, directly into two strains of E. coli. One added the genes through a plasmid, and the other integrated them directly into the bacterium's own chromosome, along with the chloramphenicol resistance gene. All of these genes code for a pathway which is able to turn that glucose into ethanol as shown in figure 1b, without a byproduct of acetate and with much higher yield than the natural process found within the E. coli genes. In fact, this method is able to produce 95% of the final products as ethanol without creating any redox imbalance, yielding 46 grams of ethanol per liter with cheaper materials than required for the natural process. Therefore, by adding a plasmid and changing a couple of genes, E. coli has been able to go from an annoying bacterium that infects our gut to a living bioethanol factory that will revolutionize today's fuel industry.